Good morning. I'm trying to start your lecture again. Today we shall discuss on plant tissue culture, which is a fundamentally important topic for plant genetic manipulation. As your course is fundamentals of genetic manipulation, I think tissue culture is uh, the basic topic. Is it visible now?
Yes, sir.
Okay. So our topic is plant tissue culture. Plant tissue culture is one of the oldest but smart, currently useful plant biotechnology, and it is done in the in vitro condition, that is in the glass, in the laboratory in control environment. Here you can see the image. Plant tissue culture, if we define it, it can be defined as plant broadly. It refers to cultivation of plant cells, tissues, organs, and plantlets on artificial medium under aseptic and control environmental condition. Here you can carefully look at, we can use any part of the plant, that is plant cells, tissues, organs, or even small tiny plantlets, and we can cultivate them in artificial medium, nutrient medium, and in aseptic condition free from germ, that is in the control uh, laboratory condition where there is no chance for contamination, as well as we can control the temperature, humidity, and light, other factors. So plant tissue culture is also called uh, an art as well as a science. And it is the art of growing experimental plants, selecting a suitable plant organ. Any organ we cannot grow, so you have to select the suitable organ or tissue to initiate cultures. Cleaning, sterilization, trimming, these are the steps, major steps we do for the tissue culture, it to a suitable size and planting it on a culture medium containing, nutrient for the growth of the plant material. And obviously, you maintain the light and other, environmental factors. What is the basic principle of plant tissue culture? This is a fundamental question because all eukaryotic organism we cannot culture. Bacteria we can culture, prokaryotic organism, but even some eukaryotic organism like fungi we can culture in the medium, like tissue culture. But if you consider human or other animal, they are eukaryote like plant, but we cannot culture, do the tissue culture, like take small part of the human or other animal and grow in the laboratory to get the whole animal or plant. Impossible. There are lots of genetic barriers. But in case of plant has totipotency. For example, embryo of animal is totipotent. This is why after fertilization, a single cell can uh, uh, develop to a whole organism. So in case of plant, as it is plant is totipotent, therefore uh, tiny parts, even a cell of a plant, can be grown and developed to a whole plant. So totipotency is the ability of a cell to differentiate and develop into a whole plant when given the correct conditions. This is because every cell has the genetic potential of the parent plant. And this totipotency allowed us to develop tissue culture based biotechnology, as well as it allows us to do genetic engineering at the cell level. So totipotency, for example, you take a plant cell, it will grow to tissue or callus. Callus is profusely developed parenchymatous cell, and then they can differentiate into plant organ or embryo and the whole plant. And let's know some important terminology used in plant tissue culture. Remember that plant tissue culture is still a powerful biotechnology. It has practical implications. Um, One of the important terms is explant. Explant is living tissue transferred from a plant to an artificial medium for tissue culture. Explant means you cannot take a portion of the plant from anywhere because the we choose the specific part of the plant which has the potential to grow in the culture medium, especially the part where meristematic tissues are there. Meristematic tissues means dividing tissue. So if you take older tissue, they do not respond smoothly. So we can take the bud tip, root tip, or even young leaves or other important shoot, leaves, root, flower even. You can take pollen or pollen grain or anther. They can be cultivated in the tube you can see here. Another terminology is, I told you before, in vitro culture. In vitro, the term comes from Latin word in vitro, which means within the glass, performing an experiment in a test tube. All these type of culture, including animal cells, in vitro fertilization is important. In animal, in vitro fertilization, even in human, sperm and ovum, can be fertilized and get the embryo, and also called in vitro fertilization. Tissue culture, in tissue culture, inclusive term, tissue culture is an inclusive term for growth of cells and tissues in a sterile environment. And the cell we use, they should have potential for dividing. Undifferentiated plant cell, plant callus, or plant tissue. Callus means, profusely grown parenchymatous cells, usually colorless. So other term, very important, micropropagation. Micropropagation means the production of whole plants from a small section of a plant called explant. So you take explant like apical bud, axillary bud, meristem, and you propagate in the culture medium and get a whole plant. And it is called micropropagation. Micropropagation is the practical application of the tissue culture. That means when you get the whole plant, you can grow it and cultivate it. In case of potato, in Bangladesh, micropropagation is very popular, and micropropagation is used for getting disease-free, especially virus-free potato seedlings. And those seedlings are potent for giving you very high yield. Usually the method, micropropagation method used by commercial tissue culture laboratories. And since a whole plant, including shoot and roots, is produced, which is genetically identical to the mother plant. This is very interesting point. Tissue culture is like a vegetative propagation method because here no sexual recombination is taken place. This is why usually the plants produced through the tissue culture, they are clonal propagation or a clone to the mother plant. This is very, very important point. And nowadays, tissue culture or in vitro culture or micropropagation are sometimes, used in the laboratory for genetic engineering. What are the major importance of plant tissue culture? The most important contribution made through plant tissue culture is the demonstration of the unique capacity of plant cells to regenerate full plants via organogenesis or embryogenesis, irrespective of their source. It can be root, leaf, stem, floral parts, pollen, anther, endosperm. So you can get organ development or embryo development and finally the whole plant. And another point is that either it is haploid, diploid, tetraploid, you can make the copy. Okay, so these are the very, very important point, especially some plants like orchid, rose, or some of the plants, they do not produce seeds, good quality seeds or enough seeds. In that case, by using tissue culture, you can make unlimited number of the seedling. And there are even banana. Banana, very important. Potato, no true seed. You can do that.
Excuse me, sir, I have a question.
Yes.
Sir, if a plant is triploid, right, and it breeds with another plant, what happens to its, like, how many chromosomes does it have then?
Uh, Haploid, diploid, triploid, any level of ploidy. When you do the tissue culture, uh, the seedling you develop through the micropropagation, they remain the same level of ploidy. Just you are making the copy. But in crossing, for example, haploid uh, actually cannot give you the flower or others. This is why haploid, plant uh, basically infertile. So we use colchicine, some chemical treatment to make the chromosome double. It is called double haploid. And diploid plant, most of the rice, and rice, maize, and wheat is hexaploid. Rice, maize, and most of the cultivated plants, they are diploid. So diploid, if you cross, then you can get the diploid. Triploid, and triploid, if you make, then interestingly, some of them can be hexaploid and most of them triploid, and you can treat them by colchicine, you can make hexaploid. For example, in case of wheat, synthetic wheat being developed. Is it clear, gentlemen?
Yes. Yeah.
Okay.
Thank you.
But in tissue culture, ploidy level never been changed. But sometimes the clonal propagation through the tissue culture you develop, sometimes some variations being observed. It is called soma clonal variation. If it is a polyploid plant like strawberry tissue culture being popular in Bangladesh. So strawberry, you cannot find true seed. So you need to propagate them by using tissue culture to get the disease-free seedling. But without tissue culture, strawberry can be cultivated by using sucker banana. But sometimes they are contaminated with the disease, a big problem. So strawberry and some other plants, sometimes when you do tissue culture or micropropagation, you may get some phenotypic variation. And this type of variation is called soma clonal variation. And soma clonal variation can be used as an asset for developing new variety. So plant tissue culture is also the best technique to exploit the cellular totipotency of plant cells for numerous practical applications, and often offers also generate technologies for crop improvement. For crop improvement, for example, haploid culture, you can culture the anther or pollen of a plant, and you can get the haploid plant. Uh, so in that case, you can make it diploid by using colchicine, one chemical which can trigger a doubling of the chromosome. Or you can even by tissue culture, you can get the triploid. Uh, a triploid and triploid to hexaploid can be produced. In vitro fertilization is another power and importance of tissue culture. Hybrid embryo rescue, variant selection, clonal propagation, that is micropropagation, that can be done. Even through tissue culture, we can combined two protoplast. Protoplast is the cell wall-less. Plant cell has thick cell wall. So if you remove the cell wall, they are composed of cellulose and hemicellulose. Then the inner membrane-bound cell is called protoplast. So you can fuse two protoplasts by tissue culture with some technique, and then culture the plant so you can develop the hybrid also. And another great importance in practical application of tissue culture is virus elimination, because you use the shoot tip. Shoot tip contains no virus, although seed was contaminated with the virus. Plant is contaminated with virus. So if you culture the shoot tip, then you can get virus-free, the seedling. And in case of potato, largely being used. So virus is a very big problem for reducing the plant growth and yield, and it causes disease. Germplasm conservation. Germplasm means some genetic materials you want to conserve. For example, you have only a single plant in your country, an extinct, almost extinct plant. Then what you can do, some tissue you can preserve and make the copy and make the plant again visible and useful. Production of industrial phytochemicals. This is another practical implications of plant tissue culture. That is, plants produce diverse medicinal compounds to defend themselves from the attack of other organisms or for their fitness to the environment. This is why some of the plants are tagged as medicinal plants. But if you want to get the medicine from the plant, you need to cut the plant, and you need huge number of plants, because most of the important medicine present in the plant in a trace quantity. What you can do, you can cultivate the tissue of the plant in a big fermenter and trigger to produce your phytochemical that you desire and extract from them. So without cutting the plant and destroying the green in the environment. So you can do that. Even we can engineer a plant to produce important medicine, and then the tissue can be cultured in the laboratory for industrial production of certain medicine or drug molecule. And this type of genetic engineering is called biofarming. Farming spelling is P-H-A-R-M-I-N-G. Biofarming means the process of developing genetically modified plant for the production of drug molecule, your important complex drug molecule, which cannot be synthesized in the laboratory, but you can get higher amount through the genetically engineered or modified plant. Other point of tissue culture importance is regeneration of plants from genetically manipulated cells by recombinant DNA technology or cell fusion or genome editing. That means we do genetic modification using recombinant DNA technology. Perhaps you know that. That is, you bring a gene of interest from other organism like Bacillus thuringiensis, BT gene, and then you combine it in the plasmid of a bacterium like Agrobacterium tumefaciens, and then grow bacterial cells with the your target plant like eggplant or brinjal to transfer the BT gene into the brinjal genome. And as agrobacterium tumefaciens can infect the plant cell and they have the TI plasmid to transfer the gene from their genome to the plant genome. This is why this bacterium has been used as a tool for vector for genetic engineering. So in that way, you can make a transgenic or GMO plant. In Bangladesh, BT brinjal, BT cotton, BT means Bacillus thuringiensis. Both cotton and brinjal, they carry a gene from the bacterium that you see. From tiny bacteria to a plant, you can transfer the gene. And in that case, all the transformation can be done at the cellular level. And then you need to get the plant. So cell you culture in the lab and get the plant through micropropagation, and that plant uh, after screening you get the genetically modified plant. Another point is cell fusion. I told you after removing the cell wall, the membrane bound plant cell is called protoplast. You can fuse two protoplast and make the hybrid if they contain different traits, and then you can culture and get the through micropropagation hybrid plant. Really interesting. And tissue culture not only recombinant DNA technology currently used CRISPR-Cas genome editing or other genome editing technique. Editing is done at the cellular level, but through the tissue culture and micropropagation, we can get the gene edited modified plant. So do you have any question? This part is very important, why tissue culture is being widely used in the laboratory as well as in the industry. Next, other importance of tissue culture, it is extensively being applied for basic studies related to plant physiology, for example, photosynthesis, nutrition of plant cell and embryos, biochemistry, cellular metabolism, morphogenesis like organogenesis or embryogenesis, phytopathology, that is plant microbe interaction, and histology, cytodifferentiation, cytology, cell cycle. That means to study about the biology of the cell and a plant cell and its interaction with the environment, even microbes and others, and fundamentals how in the cellular system functions, we need the tissue culture. Because you can culture pure cell line cell and then you can observe the biology very precisely. So biochemist, botanist, cell biologist, or plant pathologist, molecular biologist, they also use tissue culture to study the inside of the cell biology and molecular biology. Indeed, the discovery of first cytokinin is based on plant tissue culture. Scientists discovered cytokinin, a major plant hormone, based on the study of plant tissue culture. So many new molecules being developed, molecular mechanism being developed, even the pathway and processes like photosynthesis. Scientists discovered the mechanistic aspect through the tissue culture. So this is all about the importance. Now I shall try to share a brief history of the development of plant tissue culture. When we call tissue culture, we should remember the Gottlieb Haberland, a German botanist. He made the first attempt to culture fully differentiated single cells isolated from the leaves of Lamium purpureum, petioles of Echinonia, crassips, glandular hairs of pulmonaria mollissima, and stamens hairs of Triscantia in a simple nutrient solution of NOP. That is, he made a nutrient solution and able to culture the leaves, petioles, glandular hairs, and stamen hairs of some plants, and started the process of history of tissue culture. And the purpose of that experiment was to achieve divisions in these cells and obtain complete plants from them to verify the concept of cellular totipotency inherent in famous cell theory put forward by Sladen and Swan. Actually, Sladen and Swan, they gave the theory of totipotency. So you may be asked who first gave the cell theory. Plant cell theory, that is, cell theory was given by Sladen and Swan, and they proposed that uh, uh, there are cellular totipotency. That is, from a single cell, you can get the whole complex organism. And Gottlieb, he did the first experiment to get to prove the cell theory. And then another guy, a very famous, who for the discovery of auxin. Auxin is a major phytohormone. Phytohormones mean plant hormone, and he is famous for the discovery of auxin. Roger discovered the auxin and recognition of the importance of B vitamins in plant growth. Uh, gave the required impetus of further progress in the field of plant tissue culture. I shall tell you, when you culture a plant tissue in the laboratory, not only nutrient medium, you need to add appropriate amount of different phytohormones to trigger, shoot and root growth. Auxin is really the most famous phytohormone, plant hormone. And he also found that B vitamins also required for plant growth. So these are the starting point, how artificially in the, in vitro condition, you can cultivate the plant cell using indole acetic acid. Auxin is also indole acetic acid. Indole acetic acid and B vitamins. This guy, Gauthard, obtained continuously growing cultures from carrot root tissues. Okay, and then we must remember another guy, Pierre. Why? Because in the same year, White and that is Novecourt, Pierre, reported that establishment of callus culture from tumor tissues of the hybrid Nicotiana glauca. Nicotiana means the genus contain the tobacco plant. So Nicotiana glauca and Nicotiana Longsdorffia, Dorfi and carrot respectively. He found that the tumor tissues of these plants can develop the callus culture. Callus culture means profusely, from a single cell, profusely developed parenchymatous cell. Those cells are undifferentiated, no organ, just mass of the cell. So he was able to do that, and it was starting point. Even now, if you do the tissue culture, first you get the callus, and then you can get the organogenesis by using different combination of hormones. These three scientists are credited for laying the foundation for further work in the field of plant tissue culture. Yes, here are two great guys known for their method, not exactly method. They developed a culture media uh, in combination with complex nutrients, uh, and that medium is still being used. Toshio Murashige, Japanese scientist, and Folke Skog, and they developed a method called Murashige and Skog medium. Even now, MS medium, widely used worldwide industrially. You can buy those that medium from the company. They developed it. You see, during 1950s, Skoog and his co-workers at the University of Wisconsin, USA, made several major contributions towards the progress of plant tissue culture. Jablonski and Skoog, another pair, tested several plant extracts to induce divisions in mature pith cells of tobacco and found yeast extract to be most suitable in this respect. Toshio Murashige was instrumental in popularizing micropropagation, and they finally developed a medium called Murashige and Skog medium. And usually, at that time, they used the horticultural plant. Horticultural plant means garden plants, fruits, vegetables, ornaments. They are horticultural plants. So, which are the requirements for in vitro culture? So if you are asked why Murashige and Skog are famous for, Murashige and Skog are famous for their development or discovery of Murashige and Skog medium, that is MS medium, that is widely being used for plant tissue culture. Which are the requirements for, and it is also called, MS medium is called universal tissue culture medium. Which are the requirements for in vitro culture? Previously, I told you in vitro glass culture. Conditions for working in a sterile environment. You must ensure that the conditions for your working is free from any contamination, that is sterile environment, no germ, no microorganism, because in the atmosphere, lots of microorganism is there. And then defined tissue culture medium. You need defined tissue culture medium, for example, MS medium. MS medium, although it is robust, but you need to add something, hormone and other thing, to make, customized medium for your targeted plant tissue culture. And then you need an explant. Otherwise, you cannot get the tissue culture. And methods for in vitro propagation. This is very crucial point, because although tissue culture is very popular and almost theoretically, almost all plants can be cultured from the tissue, but plants to plants, even genotype to genotype, even within the same species, response to the tissue culture are hugely being varied. This is why requirements, that is the hormone, nutrient, condition, you may need to optimize and develop a method, a specific method for specific plant of your interest. And sterile work means pre-treatments. You do the experiment with all the equipments free from the contamination. So what you need to do, for example, you get a plant explant, or you are using the scissor, knife, loop, and other materials, all need to be, free from the germ. So in case of explant, you need to remove the surfacely, contaminated microorganism by treating some surface sterilizers, and your hand also need to be sterilized by using ethanol, spray ethanol and clean your hands. You must wear the gloves, mask, and obviously the clean bench, the laminar airflow cabinet you can clean, and all the tools you used, even a scissor, knife, scalpel, they need to be autoclaved and free from the microorganism and force outgrowth of auxiliary buds, remove surface contaminants by rinsing the force outgrowth of auxiliary buds, that is your explant, and remove surface contaminants by rinsing and sterilizing the solution. There are some chemical solutions, so you put the bud and remove the external solution and then wash with the distilled water, that is sterilized distilled water. Always use water, autoclaved water, and otherwise water also carries the bacteria, fungi, and other organisms. So bacteria and fungi, unless removed completely, will overgrow the explants on the tissue culture media. Why we remove the bacteria, fungi? Because when you grow the cell or tissue in the nutrient-rich media, they will these microorganisms will overgrow, faster grow than your tissue culture, and it will contaminate, and you cannot get the pure tissue culture. So sterilants used in the laboratory for the removing the microorganism from the uh, auxiliary bud or explant. We use oxidants, active halogen, heavy metal poisoning, powerful chemicals such as sulfuric acid may be used in case of seed as far as possible. Cut surface should be protected. There is always a trade-off between killing surface contaminants and killing the explant. If you do not maintain the concentration of these toxic, sterilants, then it may kill your explant. This is why, as explant, plant cell has cell wall, so they are relatively protected than the fungi and bacteria. But immediately after sterilization, you need to wash the explant in sterilized water to remove the all poisons or sterilant. Uh, and here is the different sterilants: sodium hypochlorite, calcium hypochlorite, hydrogen peroxide, mercury chloride, silver nitrate. And usually these heavy metal containing uh, materials are less, encouraged, because they are heavy metal is a pollutant. But others you can use. But sodium chloride, calcium, sodium hypochlorite, calcium hypochlorite, hydrogen peroxide, whenever you use, you need to take enough care, because they are toxic for even your cell. This is why you need to use the gloves. Antibiotics are rarely used since many are merely bacteriostatic. Massive outgrowth of cultures can result from residual bacteria or and or selection of resistance. Sometimes antibiotic use, but less being used. There are no antifungal compounds that are proven to be innocuous. So sodium hypochlorite, calcium hypochlorite, hydrogen peroxide, they are fantastic. Sodium hypochlorite is very good. And sterile environment, how can you maintain it? This is a laminar airflow cabinet. Here you can put all the devices you need to use. See, sterilized water and all other devices, scalpel, forceps, all are sterilized. And here you can see lots of holes. That means air uh, is coming out, not from outside. Air does not go out. And there is a glass cover here. So from your face, your mouth, bacteria and other microbes cannot grow. So this is the tissue culture cabinet or laminar airflow hood. We do the tissue culture. So agents used for surface sterilization, they are like this. Just a moment. Sorry, the electricity connection to my laptop was a bit interrupted. Anyway, so This is the summary of the agents used for surface sterilization of the X plant. So X plant surface may contain microorganisms. You need to remove them. And there are lots of agents, chemical agents, as I mentioned, hydrogen peroxide. Here is the dose and how long time you treat is written there. And after treatment of any surface sterilizing agent, remove the explant and wash it at least 3 to 5 times in sterilized distilled water so that you can remove these toxic substances from the explant before placing to the tissue culture medium. So schematic representation of protocol for surface sterilization. So first you collect the explant, for example, an auxiliary bud from a plant or root tip or leaf tip, and then you rinse with water for 15 minutes. First time rinse with water. And then surface sterilized, that is sterilized water, autoclaved water. And then treatment with babystin for 20 minutes, rinse with water for 15 minutes, treatment with T-pol for 15 minutes. These are, for surface sterilization. So surface sterilization, you can use other thing also. Wash with sterile distilled water, and then treatment with 0.1% mercury chloride for 2 minutes. This is, a schematic presentation using these surface sterilizing agent. But depending on your plant and availability of the surface sterilizer, you can change the chemical and even timing concentration also. But these are the major steps. Finally, sterile distilled water, you need to put your explant and completely remove the mercury chloride or other surface sterilizing agent before placing to the
Sir,
medium.
what is babystin?
Babystin also surface sterilizer, and there are many kind of uh, surface sterilizing agent people use. So ultimate target is after getting the explant from the field or garden grown plant, then you first wash with the water 15 minutes so that water is universal solvent, and also a medium for having the microorganisms. So loosely attached uh, materials, even debris, all you can clean it. And then you start, a multi-phase surface sterilization of your materials. And every step you need to wash with water. Water means, autoclaved water, pure from, free from the any germ. And finally, you completely sterilize, wash the explant, which is supposed to be free from microorganism by deionized water, that is autoclaved water. In that case, here I mentioned mercury chloride or other thing, babystin or some other T-pol. There are many, even sometimes we use uh, ethanol, instead of babystin, ethanol for short time, because ethanol can kill the surface, bacteria and fungi. For example, ethanol for 5 minutes. So you drop. It depends on what kind of materials you are using. Time also depends, because if you keep longer time, then your plant cell also be killed. You cannot kill the all plant cells. Some of the plant cells may be killed with the undesirable microorganisms, but there might be living cells after surface sterilization. Otherwise, if you put into the tissue culture medium, you cannot get the living cell. Do anyone have any question? No? Okay. Here you can see the tissue culture. This is a tissue culture incubator, different kind here in the plate and then in the bottles. So how it happens? So surface sterilization and then culture on a suitable medium. Suitable medium, for example, MS medium containing appropriate concentration of oxygen, cytokinin. Oxygen, cytokinin, they are very famous, phytohormone. Oxygen, cytokinin, gibberellin, abscisic acid, and ethylene, five most famous, plant hormone. Oxygen, gibberellin, cytokinin, abscisic acid, and ethylene. Ethylene widely being used the ripening of the fruit in nature also. So they are very famous, and in the tissue culture, oxygen, cytokinin, gibberellin, you need to use. And then when you culture medium containing some hormone, then you will get somatic embryogenesis. That is, cell will profusely grow, and first you can get the callus, and then cell differentiation will be taken place, and first globular structure, heart stage culture. First time no chlorophyll, but by changing the hormone and move to the new plate, finally you can get, here you see somatic embryogenesis taken place, but they are undifferentiated. But finally, you will get this, single plant with the root. Root shoot is necessary. Then you can remove it from one plate to other, and finally you move to the, soil containing the pot. But before moving to the seedling, to the soil containing pot, you need to keep it in a mild environment, not directly to the sunlight, because it cannot tolerate as it is cultivated in the control environment. So the process is called hardening to make it hard. So in the shade, a calm and cool environment, and then put into the soil, the root start to establish. Once it is established, then you can move to the sunlight, and it would be okay. So hardening is a process from tissue culture, tube, a seedling from tissue culture tube to the real field. You need special care and semi-control environment to make the plant harder enough to survive in the real environmental condition. In vitro culture, also called clonal propagation, and we have biggest target is disease-free seedlings in micropropagation. So potato production, reproduction by stem tuber. This is widely being used. If you asked, give an example of the practical application of tissue culture for improvement of food and nutritional security in Bangladesh, obviously you put the potato, because bumper production of potato is achieved due to the tissue culture. And now through tissue culture, microtubers are developed from the stem tuber. The potato we are eating is basically a modified stem. So from that stem, there are some eyes. You can get the bud, and that can be placed into the medium as explant, and you can get profusely used plant. And potato previously, every year we need to bring the seed from Netherlands and other countries, but now we are self-sufficient to produce potato seed through the tissue culture. Ornamentals like orchid and many other ornamental plants, even gerbera, all can be done through the tissue culture. In Joshore, Godkhali, most of the flowers are produced over there, and they also use tissue culture seedling to have high quality, disease-free flowers. So in many countries like Netherlands, Thailand, they export flowers in China producing the tissue culture. Even you can do a business, tissue culture business. One of my student from Rabia Rimi from East West University, after graduation, she started tissue culture business in Bangladesh. So strawberry meristem culture, meristem culture is very free virus and fungus free seedling, and it is popularized in Rajshahi. Professor Munjur Hussain, he actually introduced strawberry in Bangladesh when he did PhD in Japan. He brought the protocol and introduced in Bangladesh, and he has a tissue culture, company called Akafuji tissue culture, laboratory. So he's doing business, developing seedling of strawberry. And for doing an industry of tissue culture requires very less investment. Uh, Only technical know-how is important. Even a single room is okay. Rooting of cuttings, different conifer, grafting of trees, palm, apple, circumvent seed dormancy. Some seeds are dormant, usually cannot be germinated. So in that case, tissue culture is very, very nice. Banana is popular, citrus, or many other, ornamental and horticultural plants, we can do the tissue culture. So lots of examples you can find in the internet. Tissue culture is still a multi-billion-dollar business worldwide. Reasons for in vitro cultivation of plant material already I discussed, but let me review again. Control environmental conditions. We can do lots of science, discover the biology of the plant. A symbiotic orchid seed culture we can do. Metabolite production we can do. Pathogen-free materials we can develop, especially in commercial agriculture, pathogen-free seedlings, that is potato, banana, and many other crops. We can get pathogen-free seedlings. Multiplication, rapid clonal propagation. For example, from the seed, seed is a limited resource, but tissue culture is a renewable resource, never been replenished. So you can do unlimited number of plants or tissues from a single cell. Cryopreservation, some extinct plant you can preserve as a tissue in the a form of cryopreservation. Cryopreservation means to preserve some living tissues or cell at very low temperature, minus 160 degrees Celsius. At that condition, if you preserve the cell, then cell become silent, that is deep sleep, goes to deep sleep. For example, embryo, not embryo, embryo can be preserved, but the sperm and ovum can be preserved for a long time. Even human sperm can be preserved for a long time at minus 160 degrees Celsius, and this process is called cryopreservation. So, if you afraid of certain plant will be extinct soon, then you can preserve the tissues into the minus 160 degrees Celsius for future use. Genetic engineering, I told you, for recombinant DNA technology or gene editing, or protoplast fusion, you must need tissue culture. People thought in future for genome editing, tissue culture may be no need because in plant protocol development is in progress. But anyway, tissue culture is very, very important for genetic engineering, and tissue culture is very, very important for science, as I mentioned earlier, for cell biological study, biochemical study, molecular biological study of the plant. So in vitro pollination, even in vitro pollination can be done because pollen grain or anther you can culture in the uh, medium, and then you can get the haploid, and then you can do the colchicine treatment to get the full plant, and this is called double haploid technology. Double haploid technology is very, very popular and also used in speed breeding. Obviously, these terms you need to be familiarized. Collagenesis, callus formation is called collagenesis. Embryo formation is called embryogenesis. So first callus form, and then embryo form, and then organ develop. That is shoot formation is called that is organogenesis. Then rhizogenesis, root formation. So these are sequentially being arranged, and the process is triggered by different hormones. So types of tissue culture. There are several types of tissue culture. One is meristem culture, depending on different explant. Meristem culture. Meristem means the cell potential for division, especially bird or like that, elongation of dormant meristems, and embryogenesis from somatic cells. Somatic cells means cells in the organ, and organogenesis from callus directly on explant. So you can take the callus, so you can and get the plant. Adventitious shoot formation. Adventitious shoot formation is de novo development of shoot from the cell clusters in the absence of pre-existing meristems. Actually, first you will get the callus when you culture, profusely cultured, and then embryogenesis, organogenesis, and even should root rhizogenesis taken place. So factors determining the success of tissue culture. Tissue culture a bit delicate, but once you are familiar to and become expert, then it is a routine job. So species, very important. Plant species, which plant you are going to do the tissue culture. Some plants are recalcitrant, not easy to do tissue culture. So in that case, you need huge trial to optimize the condition. Genotype, even within the species. For example, rice has 10,000 genotypes in our country. So all genotypes are not easy to do tissue culture. Age of the tissue, yes. The explant you are using, the age is very important. Younger one is better. Explant size as well as type. Environmental conditions, temperature, light, day and night, lighting is very important. Composition of culture medium, this is very crucial. So all factors are genotype dependent and require optimization for each cultivar. You cannot use a certain recipe for all the plants or all the genotypes. They have the specificity, specific liking. Plant tissue culture medium contains inorganic compound, mineral nutrition. A plant, require at least 16 essential nutrient elements, and nitrogen, phosphorus, potassium, calcium, magnesium, like this, 16 or 17. So your medium must contain those inorganic compounds. And then you need to use sucrose, carbohydrates, some energy source, and then plant growth regulators, different hormones, auxin, gibberellin, cytokinins, and others, and some other miscellaneous compounds like vitamin, and make a recipe, and this recipe actually through trial and error people developed. And you can, if you start now, for example, ORCID, then you can get the published protocol for, culture media, and then you try and modify it. And inorganic compounds also, some are macronutrients, some are micronutrients. Macro means the amount requires higher amount, and micronutrients very less quantity. Micronutrients, if present high concentration, then it is toxic, even for human or other organisms. All essential nutrient element, the appropriate concentration is very important. Nitrogen requires the most because it is the essential element for DNA, RNA, protein, and other amino acid, all other compounds. Phosphorus, obviously, a component of DNA, and many other phosphoproteins and lipids, and potassium also very important. So these are the macronutrients, these are the micronutrients. Here you can see Murashige and Skoog universal tissue culture medium. I told you this is the combination. Imagine how they made this combination. Really was very difficult, but this is still being used, Murashige and Skoog medium. And here pH is also, when you make the medium, you need to check the pH, the acidity and alkalinity of the medium. So pH 5.5 after autoclaving is the best. And autoclave, how many minutes? 15 minutes, 121 degrees Celsius is necessary. So when you make the tissue culture medium, you must do autoclave. And then you get the autoclave media free from all the germs and all the chemical, nutrient compounds. Here you can see lots of vitamins and other, even sucrose being used. I mentioned a typical plant growth regulators used in tissue culture: auxin, cytokinins, gibberellic acids. What auxin do? Auxin induce callus formation and also induce root formation, rhizogenesis, calogenesis, or like that. So cytokinin, induce shoot formation or embryos development. Gibberellic acid, cell growth and elongation. So these are their purpose. Appropriate concentration of and combination of these phytohormones are essential for successful tissue culture. Here you can see the structure of different auxins, chemical structure. So indole 3 acetic acid, as well as naphthalenic acetic acid, or 2-AD, IBA, all are auxins. So you can use any one of them or a combination. Auxin is very interesting. Auxin transport, it is produced and then transport through the plant. For example, you take this bud, and here, if you put the auxin, auxin can pass through. Always, you see, going downward. But auxin cannot go upward. Very interesting. And it is been because it is produced in that root during sunlight and goes downward to the root. And apical dormancy, here you can see how you can get the, how to say, the bud for tissue culture. And apical dormancy of any plant is governed by the auxin. Bud formation is regulated by the auxin. And auxin also adventitious a root formation. Here you see no root, but if you put auxin, then root formation taken place. So auxin, a plant growth regulator, and its physiological function includes phototropism. Phototropism means movement, preferential movement of a plant to the sunlight, that is photo light. In case of sunflower plant, if you carefully look, always it faces to the sunlight. All the plants, show more or less phototropism. And here the main trigger molecule is auxin. Apical dominance cell division, cell differentiation, initiation of embryos and organ. And synthetic oxygen, commercially being available, are often more effective than the natural oxygen. Cytokinin, these are the structure of cytokinin, basically purine type compounds, and naturally they are gelatin or other compound, but benzyl, aminopurine, or uh, thidiazurab, all are bind to a specific receptor and do the same job. So cytokinin also you can buy from the market. Um, what is the main function? Adventitious shoot formation, inhibition of root formation. That means if you use high concentration of cytokinin, then your tissue culture plant cannot produce the root. This is why oxygen cytokinin ratio is very crucial. Cell division, callus formation and growth, stimulus and outgrowth of auxiliary buds, inhibition of leaf senescence. Leaf drop, in winter, many plants drops the leaves. It is called senescence, but cytokinin keep the leaf young and not allowed to drop. Here you can see oxygen and cytokinin act synergistically. This is the medium, you put the X plant. Here, oxygen, you use indole acetic acid, cytokinin, you use kinetin. So if you use 3:0.2, the oxygen and cytokinin, you can find only callus, continuously callus, profusely growth of plant tissue, parenchymatous tissue. But if you put 3:0.02, here you can see root formation took place. But here, if you change only the shoot formation, and if ratio is like that, no indole acetic acid, no growth. This is really fantastic how oxygen and cytokinin act synergistically to in the tissue culture condition. Really amazing. So effect of cytokinin and oxygen on senescence, different ratio is shown here. Similarly, oxygen and cytokinin high, low, you need to make a balance to get your desired tissue culture plant. Another phytohormone I told you, gibberellic acid, synthesized from mevalonate pathway, and it is very important for fruits and tubers. And only some form of biologically active gibberellic acid are important. Dramatic effects on cell elongation. Cell elongation. If you spray gibberellic acid in grapevine orchard or some fruit orchard, you can find bigger fruit. Promotes cell division in combination with oxygen, that is indole acetic acid. Effect on seed germination. Sometimes some seeds cannot be germinate and become dormant. Gibberellic acid can wake up them. Improves fruit set, fruit growth, fruit maturation, and fruit ripening. Promotes flowering. Sometimes floriculturist, they use gibberellic acid spray for promoting the flowering. So effect of gibberellic acid, here you can see dwarfing mutant, wild mutant. So if you use gibberellic acid high, you can make the dwarf plant also. So we can use a different medium, protoplast culture, suspension culture, homogeneous distribution of nutrient, oxygen deprivation. Lots of laboratory practices are required to do the tissue culture. Sometimes cell culture in a big bioreactor, you need only the callus, no need the full plant for drug discovery or like that. And liquid medium we used in the tissue culture, semi-liquid actually. We use the agar medium, Gelan gum, solidified media on the tube or bottles. Gelling agent we used in the tissue culture. Red algae produce the polysaccharide called agar. And agar you can melt easily and solidify. They do not produce any nutrient, but they are good for root penetration and growth. And Gelan gum a bit softer. This is also exopolysaccharide produced from the bacteria, and the previous one, agar, is produced by the seaweed. So Zealand gum also a solidifying agent for the nutrient medium we can do. So hybrid preparation propagation also can be done by using tissue culture in case of asparagus or like that. Hybrid bigger, very important. Interspecies crossing. Sometimes crossing different species cannot be done, but protoplast you can fuse and then grow, and you can get different colors of flower. Mutations can be done. For example, you do the tissue culture and then you expose them to gamma radiation and other radiation. You can modify the genetic code, that is, do the mutation, and you can get different, phenotype. And if it is liked by you, lots of flower, a short plant, and it is commercially important, then you can select this one. That means tissue culture can be used in the breeding. Industrial application, I told you, in the big fermenter reactor, for example, chiconin is produced from the plant, and many other, pharmaceutically important compound can be produced by culturing the plant cell. And many, plants that cannot produce, viable seeds, we can do the tissue culture, in vitro, oil palm, and many other plants. So this is all about the lecture. And now I welcome you for a discussion. So we can discuss 10 minutes. That would be fantastic. So I welcome all of you for participation in the discussion. Otherwise, uh, one-way lecture is not good.
Sir, could you please explain advantageous shoot formation? Please define the term. I didn't understand.
Adventitious shoot formation. For example, you cut a stem, and then if you put into the soil, it cannot grow because it has no root. But by putting the certain medium, it is, a vegetative plant, part of the plant, you can get the root. The root usually generate from the seed, but here adventitious root, not the original root. From the stem, root can be generated. And in many plants, even if you cut a stem, put into the soil, adventitious root is formed. There are two types of root. Root is, one is main root system. For example, a tree, there is a principal root goes down, and there are some branch root. But adventitious roots are not the primary root. They are, like around the stem, they will generate and get the nutrient. But adventitious root is a bit different from the root generate from the, for example, dicotyledonous plant and other plant. But obviously, they do the function of holding the plant as well as the uptake the nutrient. So adventitious root generation can be triggered by applying the hormone, phytohormone. Okay. Amina, is it okay? Next. Any more question?
স্যার আমার একটা কোশ্চেন ছিল স্যার।
Yes, sir.
স্যার একটা স্লাইডে ইম্পর্টেন্স অফ প্ল্যান্ট টিস্যু কালচারে অনেকগুলো পয়েন্ট ছিল স্যার। স্যার ওখানে একটা পয়েন্ট লেখা ছিল স্যার যে সোমাটিক হাইব্রিডাইজেশন। স্যার ওটা একটু পয়েন্টটা বোঝাতে পারবেন? সোমাটিক
Yes, আমি বোঝাতে পারবো, এবং এটা তোমাদেরকে বলছি। হাইব্রিডাইজেশন হচ্ছে একটা প্রসেস যেখানে দুটো ডিসিমিলার প্ল্যান্টের মধ্যে আমরা ক্রসিং করি। ক্রসিং মানে কি? পোলেন, একটা উদ্ভিদের মেইল সেক্স অর্গানটা নিয়ে ফিমেলের স্টিগমার মধ্যে দিলে ফার্টিলাইজ হয়। উদ্দেশ্যটা হচ্ছে যেটা থেকে মেইল পোলেনটা নিয়ে আসছি, এইটার বৈশিষ্ট্যকে আমি যেটা স্টিগমার মধ্যে দিয়েছি এইটাতে ট্রান্সফার করতে চাই। পরে যে সিড প্রডিউস হবে, এই সিডের মধ্যে উভয় প্ল্যান্টের বৈশিষ্ট্য থাকবে, এটাকে বলে হাইব্রিড। ঠিক আছে, হাইব্রিড একটা জেনেটিক রিকম্বিনেশন হচ্ছে, কিন্তু কোন জিন যাচ্ছে এটা তুমি শিওর না। তখন তুমি বাছাই করে ঠিক করবা যে কোনটার, কিছু হাইব্রিড আছে তোমার ডিজায়ার্ড ফেনোটাইপ দিবে, ট্রেড দিবে, মনে করো হাইট বা ফ্লাওয়ার খুবই বেশি দিচ্ছে, এগুলোকে তুমি সিলেক্ট করবা। তো এই হাইব্রিডাইজেশনটা ক্লাসিক্যালি মনে করো যে 100 ইয়ার্স লোকজন এরকম, গ্রেগর জোহান ম্যান্ডেল তোমার এই হাইব্রিডাইজেশন করেছে পি প্ল্যান্টের মধ্যে, সেই জন্য জেনেটিক্স এর ফাদার বলা হয়। কিন্তু আমরা এই হাইব্রিডাইজেশন সোমাটিক লেভেলে করতে পারি, ওইটা তো হচ্ছে তোমার এই সেক্সুয়াল রিকম্বিনেশনের মাধ্যমে, অর্থাৎ রিপ্রোডাক্টিভ অর্গানের এই রিকম্বিনেশনের মাধ্যমে হাইব্রিডাইজেশন, যেটা ন্যাচারালি পলিনেশন যে হয়, এইটাও কিন্তু একটা ন্যাচারাল হাইব্রিডাইজেশন হয়। কিন্তু আমরা যদি দুটো উদ্ভিদ আমরা জানি, একই স্পেসিস, মনে করো একটা অ্যারোমেটিক রাইস আর একটা হচ্ছে নন অ্যারোমেটিক, আমরা এই দুটোর সেলের ওয়ালকে সরিয়ে দিয়ে যে প্রোটোপ্লাস্ট পাবো, এগুলো হচ্ছে সোমাটিক সেল। এদেরকে ফিউজ করা, ফিউজ করে তোমার যে নতুন টিস্যু কালচার করে প্ল্যান্টটা পাবা, এটাকে বলা হয় সোমাটিক হাইব্রিড। মানে একটা রিকম্বিনেশন হবে এবং এই প্লান্ট থেকে তুমি এই বৈশিষ্ট্যের এই সংযোজন পাবা অথবা রিকম্বিনেশন পাবা, এটাকে সোমাটিক হাইব্রিডাইজেশন বলা হয়। এটা অ্যাডভান্স লেভেল অফ টিস্যু কালচার, যেখানে আমরা নিউ ভ্যারাইটি ডেভেলপ করতে পারি। ঠিক আছে, কিন্তু তুমি শিওর না কোন জিন যাচ্ছে, যেমন আমরা রিকম্বিনেন্ট ডিএনএ টেকনোলজি, জিন এডিটিং একেবারে প্রিসাইজলি করছি। এখানে যখন তুমি এরকম ফিউজ করবা, অনেকগুলোর কম্বিনেশন হবে এবং এইটা যে হাইব্রিডটা হবে, এইটা তোমার ডিজায়ারেবল হতে পারে, আনডিজায়ারেবল হতে পারে। তো এখানে যেটা করতে হয়, অনেকগুলো থাউজেন্ডস অফ প্লান্ট করে এখান থেকে বাছাই করতে হয় যে কোনটা তোমার ডিজায়ারেবল হয়েছে। ঠিক আছে, অনেক সেলের যখন এরকম ফিউশন করা হয়, প্রোটোপ্লাস্ট ফিউশন। ঠিক আছে,
জি স্যার।
ওকে, থ্যাংক ইউ শামিন। আর কারো প্রশ্ন?
Sir, sir, there are various culture mediums such as inorganic compounds, plant growth regulators, and miscellaneous. And I want to know which compounds are included in miscellaneous medium.
মিসসেলেনিয়াস, এটা খুব ভালো প্রশ্ন করেছো। বিভিন্ন রকমের ভিটামিন ইউজ করা হয় মিসসেলেনিয়াসের মধ্যে। তোমার যেগুলো এমনিতে এসেনশিয়াল নিউট্রিয়েন্ট এলিমেন্ট না, মনে করো সেলেনিয়াম ইউজ করা হলো, এটা এসেনশিয়াল নিউট্রিয়েন্ট এলিমেন্ট না, অথবা কোনোটাতে সিলিকন ইউজ করা হলো, অর্থাৎ এইগুলো হচ্ছে ডিফাইন্ড নিউট্রিয়েন্ট মিডিয়াম না। কিন্তু রিপোর্ট আছে যে তারা টিস্যু কালচারের এফিসিয়েন্সিকে বাড়াতে পারে, গ্রোথকে বাড়াতে পারে। এইরকম অনেক কিউরিসিটি নিয়ে এই লোকজন এগুলো ট্রায়াল এন্ড এরর করে, খুব ট্রেস মিসসেলেনিয়াস অলওয়েজ খুব ট্রেস কোয়ান্টিটি, তোমার এইটা একটা মলিকুলও হতে পারে অথবা একটা এলিমেন্টও হতে পারে। হ্যাঁ, এটাকে ট্রেস কোয়ান্টিটি অ্যাড করলে দেখা গেল এটা একটা তোমার টিস্যু কালচারের সাকসেসকে বাড়িয়ে দিচ্ছে। ঠিক আছে, থ্যাংক ইউ। এনি মোর কোশ্চেন?
Okay, sir.
থ্যাংক ইউ। আর কারো কোনো প্রশ্ন আছে কিনা? তোমরা সবাই একটা প্রশ্ন করার চেষ্টা করো। এখানে 33 স্টুডেন্টস অ্যাটেন্ড করছে, 32। যার মাথায় আছে এখন তো তুমি ফ্রি, ক্লাসরুম না, একটা প্রশ্ন করো। ইভেন প্রশ্ন তুমি চ্যাট বক্সও দিতে পারো। আমি আরো তোমাদের জন্য 5 মিনিটস কোশ্চেনের জন্য রাখছি, কারণ কোশ্চেন ছাড়া এটা ইফেক্টিভ হয় না যতক্ষণ পর্যন্ত তুমি ইন্টারেক্ট না করছো। অনেক জিনিস আছে খুব ব্রিফ ফর্মে বলা হয়। টিস্যু কালচার একটা বড় সায়েন্স এবং এখানে ডজনস অফ বুক আছে।
স্যার আমার একটা কোশ্চেন ছিল স্যার।
হ্যাঁ, বলো।
স্যার ওই যে একটা স্লাইডে মনে হয় লেখা ছিল ওই যে স্যার ক্লোনাল প্রোপাগেশন আর যে মাইক্রো প্রোপাগেশন আর কি ইন্টারচেঞ্জলি ইউজ করা হয় স্যার, এটা কেন? এটা সেম প্রসেসটা কি সেম কাইন্ড অফ?
দাঁড়াও, আমি স্লাইডটাতে যাই, ঠিক আছে?
স্যার আমার এক্সাক্ট স্লাইডটা মনে নেই ওখানে ক্লোনাল প্রোপাগেশন।
তুমি কি এখন স্লাইড দেখতে পাচ্ছ এগুলো যে আমি মুভ করলে?
জি স্যার দেখা যাচ্ছে।
আচ্ছা, আমি মুভ করি, তুমি বইলো কোনটাতে।
স্যার ওই যে ধরেন ইম্পর্টেন্স অফ পিটিসি তে ধরেন ওখানে লেখা আছে ধরেন ক্লোনাল প্রোপাগেশন ব্র্যাকেটের মাইক্রো প্রোপাগেশন লেখা।
এই যে এখানে দাঁড়াও,
হ্যাঁ এটা এটা স্যার।
ক্লোনাল প্রোপাগেশন
আর
আর
মাইক্রো
মাইক্রো
প্রোপাগেশন।
প্রোপাগেশন।
জি স্যার এটা কি?
মাইক্রো প্রোপাগেশনও এক ধরনের ক্লোনাল প্রোপাগেশন। ক্লোনাল
আচ্ছা
মানে বুঝছো? ক্লোনাল হচ্ছে ক্লোন, মানে ডলির ক্লোন তো তোমরা জানো, মানে
আচ্ছা
একেবারে কপি করা। এটা যেহেতু ভেজিটেটিভ পার্ট থেকে নিয়ে করা হয়, এখানে তোমার সেক্সুয়াল রিকম্বিনেশনের কোনো সুযোগ নেই, কাজে এটা প্যারেন্টের মতো হয়। আর ক্লোনাল প্রোপাগেশন উই ক্যান কল মাইক্রো প্রোপাগেশন। মাইক্রো প্রোপাগেশন এবং টিস্যু কালচারের পার্থক্য আছে, এটা তোমাদের মনে রাখতে হবে। মাইক্রো প্রোপাগেশন হচ্ছে যখন আমরা টিস্যু কালচার পদ্ধতি ইউজ করে প্লান্টকে প্রোপাগেশন করি, অর্থাৎ সিডলিং তৈরি করি। যেমন আমরা পটেটোর একেবারে ফিল্ড লেভেলে ফার্মার্সকে আমরা সিড দিচ্ছি, অথবা বানানার সিড দিচ্ছি, অথবা স্ট্রবেরির সিড দিচ্ছি। ফ্রম টিস্যু কালচার, ফ্রম সেল কালচার ল্যাবরেটরি পার হয়ে হার্ডেনিং করে ওই ম্যাটেরিয়ালসটা যখন কমার্শিয়ালি এই এগ্রিকালচারে ইউজ করা হয়, তখন এটাকে মাইক্রো প্রোপাগেশন বলে। এর মানে আমরা রাখতে চাই যে ভ্যারাইটিটা ডেভেলপ হয়েছে এক্স্যাক্টলি সেম কপি আমরা ফিল্ডে নিতে চাই, আদারওয়াইজ সেম ইল্ড হবে না। কিন্তু ন্যাচারালি আমরা যখন প্লান্ট গ্রো করি, তুমি জানো পলিনেটর এসে তোমার একটার পোলেন যখন আরেকটা পোলেনে ডিসিমিলার বৈশিষ্ট্যের একই স্পেসিসের যখন ক্রসিং হয়ে যায়, তখন জেনেটিক রিকম্বিনেশন হয়ে পিউরিটি থাকে না, মানে মাদার প্ল্যান্টের পিউরিটিটা থাকে না। সেই জন্য কয়েক বছরের মধ্যে একটা ভ্যারাইটি রিজেনারেট হয়ে যায়। কিন্তু মাইক্রো প্রোপাগেশনে তোমার পিউরিটিটা থাকে, সেই জন্য এটা ভ্যারাইটি রিজেনারেশনের কোনো সুযোগ নেই। থ্যাংক ইউ, ভালো প্রশ্ন। আর কারো প্রশ্ন আছে কোনো স্লাইডের উপরে?
স্যার একটা কোয়েশ্চেন ছিল।
ইয়েস।
ওই যে অক্সিন আর সাইটোকাইনিনের যে কনসেন্ট্রেশন ডিফারেন্সের কারণে ক্যালাস রুট আর শুট ফরমেশন হয়, ওইটা স্যার কনসেন্ট্রেশনটা ডিফারেন্ট হওয়ার কারণে কেন হলো ওই স্পেসিফিক কনসেন্ট্রেশনটা?
হ্যাঁ, আমি তোমাকে দেখাই, তাহলে তুমি আরো বুঝবা। ইয়েস, এখানে, তাই না?
এটা স্যার, হ্যাঁ।
হ্যাঁ, মানে অক্সিন এবং সাইটোকাইনিন তাদের নিজস্ব রুল আছে, এবং তারা কম্বাইনও একটা রুল আছে। হরমোন, তোমাদের আমাদের শরীরে যত হরমোন আছে, হরমোনগুলো এক হচ্ছে ইন্ডিভিজুয়াল হরমোনের ফাংশন আছে, আবার কোঅর্ডিনেটেড ফাংশন আছে। যে কোনো একটা হরমোন যখন ডিস্টার্ব হয়, হরমোন বায়োসিনথেসিস, তখন অন্যটার ইফেক্টও তোমার প্রবলেম হয়। তো হরমোন বাইন্ড করে স্পেসিফিক রিসেপ্টর দিয়ে, এরপরে একটা জিন এক্সপ্রেস করে প্রোটিন তৈরি হয়, এবং আলটিমেটলি আমরা ফেনোটাইপ দেখি। তো এখানে অক্সিন এন্ড সাইটোকাইনিন তারা সিনার্জিস্টিক্যালি ওয়ার্ক করে, কাজে এরা ন্যাচারালি প্ল্যান্টের মধ্যে একটা সার্টেইন কম্বিনেশনে থাকে। তা আমরা তো এক্স্যাক্টলি ওইটা জানি না। সেই জন্য টিস্যু কালচারে আমি যেহেতু আমার ইচ্ছা অনুযায়ী প্ল্যান্টকে খুব ফাস্ট গ্রো করতে চাই, সিডলিং তৈরি করতে চাই, সেই জন্য এই কম্বিনেশনটা নিজেরা টেস্ট করে বিজ্ঞানীরা দেখেছে যে কোন প্ল্যান্ট স্পেসিস এবং জেনোটাইপে কোন কম্বিনেশনে তোমার রুট এন্ড শুট গ্রোথ অপটিমাম হবে। হ্যাঁ, কাজে এই হরমোনের ক্ষেত্রে কোনো একটা হরমোন যদি হায়ার কনসেন্ট্রেশন হয়, তখন এইটা তোমার যে পারফর্ম করার কথা ছিল তার চেয়ে ক্ষতিকর করে। যাদের থাইরয়েড, অনেকের হাইপার এবং হাইপোথাইরয়েডিজম আছে, আমাদের থাইরয়েড গ্ল্যান্ড থেকে যে থাইরক্সিন রিলিজ হয়, এটা হাইপার হইলেও অসুবিধা, বেশি হইলেও অসুবিধা, কম হইলেও অসুবিধা, এবং সাংঘাতিক রকমের ফিজিওলজিক্যাল ডিস্টারবেন্স হতে পারে। হিয়ার এখানে দেখো, এখানে কোনো এই এই অক্সিন দেয় নাই। সেই জন্য দেখো শুধু সাইটোকাইনিন অ্যালোন তোমার এই কোনো গ্রোথকে স্টিমুলেট করতে পারে না। এটা দিয়ে বোঝাচ্ছে তারা অনেকটা কোঅর্ডিনেটেড ম্যানারে কাজ করে। আমাদের শরীরের সবগুলো হরমোন কিন্তু এরকম। ক্লিয়ার?
ইয়েস স্যার।
আইডিয়াল কন্ডিশনে কোনোটার পরিমাণ যদি একটু বেশি হয়, এখানে দেখো এখানে এইটাতে রুট শুধু হয়েছে, কোনো শুট হয় নাই। কারণ হচ্ছে এই শুট হওয়ার জন্য যে পরিমাণ কাইনিন দরকার ছিল তার চেয়ে কম সাইটোকাইনিন। যখন এই সাইটোকাইনিনের পরিমাণ বাড়িয়ে দিয়েছে তখন দেখো শুট হয়েছে, কিন্তু এখানে অক্সিনের পরিমাণ সাংঘাতিকভাবে কমিয়ে দিয়েছে। দেওয়ার কারণে তোমার এই রুট হয় নাই। কিন্তু যখন অক্সিনের পরিমাণ বাড়িয়ে একটা অপটিমাম হবে তখন শুট এবং রুট দুইটাই হবে। ঠিক আছে?
ইয়েস স্যার।
আর কোনো প্রশ্ন? দুইজন শুধু প্রশ্ন করেছে। সাফায়াত, আরিফিন, কোনো প্রশ্ন করতে চাও?
না স্যার, এটাই ছিল।
আচ্ছা, আর আর কারো কোনো কোশ্চেন? আমি তো চ্যাট বক্সে কোনো কোশ্চেন দেখছি না। যদি কোনো কোশ্চেন না থাকে তাহলে আমরা এখানে শেষ করি, এবং তোমাদের সাথে আগামীকাল আশা করছি 6:30 দেখা হবে। আগামীকাল জাপানিজ ল্যাঙ্গুয়েজ প্রফিসিয়েন্সি টেস্ট আছে। 13,000 স্টুডেন্ট পরীক্ষা দিবে আমেরিকান ইন্টারন্যাশনাল ইউনিভার্সিটিতে। ওখানে ম্যানেজমেন্ট টিমে আসি। 2টা থেকে আশা করি তোমার ওখানে টাইম লিগে বুঝতে পারবো। ততক্ষণ পর্যন্ত সবাই খুব ভালো থেকো।
আসসালামু আলাইকুম স্যার।
ওয়ালাইকুম আসসালাম।
আসসালামু আলাইকুম স্যার।
ওয়ালাইকুম সালাম।